I hope that you guys are having a good start to your day. Um, Today we're going to go over some stuff about protein function. Um, but actually, I'm going to kind of do my lesson plan kind of in reverse. Because the first thing that I wanted to bring up is the exam that you guys have on Tuesday. Now, I'm getting my tablet because I was writing down some stuff on a piece of paper, but now I'm going to write on the PowerPoint. I'm going to add a slide though, nice blank slide, so I can use that. Um, and share. Boom. Okay. So a couple of things that I wanted to talk to you about. And if you can't hear me or anything, please add to the chat, or if you wanted to, ask a question, you can add to the chat. Um, the first thing is that you guys have exam number 2 on 10/8, October 8th. Um, this exam is going to be covering amino acids and the classification of amino acids. Now, with that said, I mentioned the other day that I was going to get learning objectives put together. And what I decided to do instead of learning objectives was to speak generally about learning objectives here. And then I thought it would be much more effective and maybe a better use of your time if I gave you a practice exam. Because in my mind, learning objectives at best are, you're going to use them and try to formulate some sort of question. And so it's almost like a guessing game of like, what's Dr. Gray going to ask or what's he going to ask? Um, I decided to just go ahead and cut through that and give you a practice exam that has questions that are very similar to the questions that you'll get on your actual exam. Now, to make this practice exam as similar to and as worthwhile as possible, I've formatted it basically the exact same as your actual exam. Um, I think I ended up with more than 25 questions on the practice exam. I think I have 29 questions. Um, but every question is multiple choice, much like your actual exam. Your actual exam, so I'll make a little chart here, actually. I'll do a practice exam versus actual exam.
Yeah, I also have been sitting on one by one, so
Practice exam is like 29 questions. Actual exam is 25 questions.
yeah, well,
Practice exam is multiple choice. Actual exam is multiple choice. Now, on your practice exam, there's 5 questions that are exam 1 stuff. So your exam 1 stuff was functional groups, Lipinski's 5 or rules of 5. Um, and your actual exam, there is going to be 5 questions that cover exam 1 stuff. Now, the big reason for that is because your exam 2 score can replace your exam 1 score. Um, so that's one factor. It will replace it only if it's higher. Your practice exam has roughly 24 questions that are exam 2 stuff.
oh, okay, but it
Your
doesn't.
actual exam has exactly 20 questions of exam 2 stuff. Now, what does this exam 2 stuff consist of? Well, it consists of a couple of different things. And I'm actually just going to have these combine into one list because they're going to be identical. Um, your exam 2 stuff consists of amino acid, 1-letter codes, as well as three-letter codes. And being able to convert a three-letter code to a one-letter code, and a one-letter code to a three-letter code, and a full name to a one-letter code or a three-letter code. In addition to that, I'll say, I'll label this as FN, full name. In addition to that, amino acid classifications. And what I mean by that are polar, polar positive. Actually, let me go ahead and make that polar positive, polar negative. Circle those, and polar, put a 0 for uncharged, as well as non-polar. Okay, so those are big factors for your exam that's upcoming. Um, Make sure that you know how to do those. Make sure that you know how to classify an amino acid by, knowing its name or knowing its structure. In addition to that, did you have a question, Bella?
No, I'm so sorry. I didn't
Oh,
know I unmuted myself.
no worries. Um, in addition to that, your interactions. So I'm going to label it as R group interactions. So what we ended last class with, where we were talking about, um, they were the ionic interactions, hydrogen bonds, covalent interactions, and I believe that it was, sorry, I have to look back at my PowerPoint to see the exact term, because there's a number of different terms that can be used for this. I just want to make sure that I'm using the right one for our class. It's either dispersion forces. Let's see. Yeah, there we go. Okay, let's see. Where were we? Yeah, dispersion forces. Yeah, the last one was dispersion forces. So these four big things, and one of the things that we did last time was we looked at every amino acid R group and we said, based on their single letter code, which amino acids, and this is on the marked up PowerPoint that I posted after last class, um, which amino acids are likely to participate in ionic interactions. Aspartic acid and glutamic acid. Those are negative residues, or those are negative, polar negative amino acids. But then lysine, arginine, and histidine, those are polar positive residues. So those are great examples of amino acids that are going to participate in ionic interactions. So make sure that you study those lists. And just to clarify what I'm referencing there, I'm going to share, I'll switch my share for a second. So amino acids that are likely to participate in dispersion forces or interact through dispersion forces, disulfide bonds, um, ionic interactions, and hydrogen bonding. Those are all our likely candidates. I'm referencing these single letters up here. So NQTDESY or TEDSY and so on. Make sure that you know those because I'm going to ask questions about those. Um, so that was one of the things that I wanted to mention. This practice exam is there. You will get feedback on that practice exam. So if you complete it, oh, when you complete that practice exam, you'll get a score. And the questions I think are 4 points apiece, just like on your actual exam. But like I said, there's more than 25, so the total will exceed 100. But that is not a grade that's going to be recorded for anything. If I were you, I think what I would do is go through and take the exam and see what I got. I basically do the math and say, okay, well, I got, 4 out of 29 right. Okay, I probably need to redo this. Now, what I'm going to do is I'm going to take the exam with my notes open. Even though you're not going to be able to do that on your actual exam, I do that so then I can find the reference points. So look at the PDFs and say, okay, well, I can find what this is referencing. And then maybe on your second go around, you get a 24 out of 29. I'd be like, okay, well, that's good. I've improved. Um, he said lots of the questions focused in on this particular area. I'm going to study that more for the actual exam. That's what I would strongly advise. Some of the questions between the practice exam and the actual exam, it's just a matter of changing a couple of words. For instance, if there was a question that said on your practice exam, if there was a question that read, which of the following is a positive amino acid? Well, then I could change that question very easily and make it either which of the following is not a positive amino acid. Alternatively, I could just say which of the following amino acids is negatively charged. So I want you to use that practice exam to your advantage. I think that yeah, it's there. It's going to be available all, you know, it's up indefinitely. You can take the practice exam. I'm struggling with the word practice today. I need to practice saying the word practice. You can take that exam 100 times if you want. The only parameters that I've set up is that um, it is on a time limit. And that time limit is 80 minutes because I wanted to give you um, a situation or a scenario just like your actual exam. So your actual exam is going to be 80 minutes. Um, One other detail is that after you take the exam, you can go and look at what are the correct answers, what are the incorrect answers. If you don't want to look at the correct answers, then just go and try the exam again, resubmit it. And then you can always look at the questions and the correct answers after you take it, the second or third or fourth time. But there's what I think is a pretty even distribution of all of our day's discussions. But there is definitely an emphasis on like amino acid, single letter and three-letter codes. So I would definitely make sure that I know those. And those I think many of them are very easy. Um, and then as a follow-up to that, you can also just like that's really easy to do flashcards. Um, the process that I would go through for learning my amino acids is first get the names, and I would divide my names into polar and non-polar. And when you divide your amino acids into polar and non-polar groups, effectively you're at like there's like 10 polar and 10 non-polar. So you've taken this group of 20 molecules, 20 names that you have to learn. And now really you only have to learn 10 or two groups of 10. It just makes it easier to kind of process that information. But that practice exam is up, and that really hits on all the learning objectives that I want you to know about for this upcoming exam. If nobody has any questions, well, actually, I'm going to go ahead and proceed. If you have any questions, please don't hesitate to either, send it in chat, either as a direct message or a message to everyone, or, we can talk after class or sometime tomorrow or anything. Um, I'll probably have some office hours tomorrow. I usually am able to do them a little bit after lunchtime. So I'll post a Zoom announcement, hopefully 15, 20 minutes before and say, hey, I'm going to be available 1 to 1:30. Drop by if you have any questions. Um, if I had to guess, the people who do the practice exam are probably going to do better on the actual exam than the people who do not do the practice exam. So I would absolutely do this practice exam. There's, it's, yeah. I I I feel like I'm making that as yeah, that's a very strong push. Do the practice exam, study the practice exam. That's how you should study for this exam for exam number two. Okay, well, I'm going to proceed because what we're talking about is within the field of biochemistry. And we're talking about protein ligand complexes. Now, one of the things that's a little bit of a, I don't know if it's a jump necessarily, but a protein is a big molecule made up of amino acids. We talked about our different levels of protein structure, your primary, secondary, tertiary, and quaternary structure. Your primary structure is stabilized by peptide bonds, also known as covalent bonds. The secondary structure is stabilized by backbone hydrogen bonds. The third or tertiary structure is stabilized by R group interactions. And the fourth level, the highest level of protein structure, the quaternary structure, is when multiple proteins kind of fold together to make one complete unit. Now, a ligand, some people will say this word ligand, and some people get very, very upset and territorial, and they say it's ligand, it's not ligand. Um, I'm not going to lose any sleep however you say it, and I don't think that there's one way that's more correct than the other. But a ligand is a molecule that a protein binds to. And these protein ligand complexes are a very important part of the study of biochemistry. Now, proteins are effectively the workhorses or the workers of the body. They're responsible for the chemical reactions that occur within the body, as well as they provide structures that make up cells. Um, there's one, or there's, okay. Um, I'm, I mean, okay. So whenever I think of proteins, one of the things that I think of is whey protein. Now, there's another protein that if you've ever watched one of those TV shows, one of those like plastic surgery shows or anything to do with Kim, Chloe, and Kylie, Kim, Chloe, Courtney Kardashians. There's a protein that they mention periodically, and it's one that they get injections of. Does anyone know what I'm thinking of? It's a structural protein, and it ends with GEN or gen. Some people put it in their coffee also. It's a protein that makes your fingernails really strong. It's also known as collagen. So collagen is a structural protein, and it basically gives, your skin and other parts of your body like structural integrity. And that's one of the things that proteins do. Now, there are three major functions that proteins carry out. One is proteins can be enzymes. Enzymes that enzymes are proteins that catalyze chemical reactions. Now, another thing that a protein can do is it can participate in cell signaling and ligand binding. The binding of one ligand to a protein will cause a signal cascade of protein activity as a response. Now, this phrase signal cascade, think about that as like signal amplification. Um, I always, whenever I think of signaling cascade or signal amplification, it's almost like how a rumor works, or like a secret works. One person says it to their best friend, and they're like, "Well, I'm only telling my best friend this deep secret." And then all of a sudden, that best friend tells one of their best friends, who tells their best friend, who tells their best friend, who tells their best friend. And all of a sudden, you've gone from, oh, well, only one person in the world knows that secret to now two people to thousands of people know this secret. And so that's what that's the way that a signaling cascade kind of works. It's taking a minor signal and kind of amplifying it, blowing it up. Now, the binding of one ligand to a protein will cause this. Another role that proteins play is they provide structure. There are structural proteins, collagen being one of them. Now, I'll give a bonus point on the next test for anyone who can say another structural protein. I'll give you a hint. It starts with a K and it ends with a tin. Keratin. Keratin is a structural protein. And, that and other structural proteins, they kind of hold everything together. So, that said, these three major roles of protein functions we're going to talk about over the course of this semester. We've got enzyme catalysis, we've got cell signaling and ligand binding, and then we've got structural proteins. Now, with enzyme catalysis, essentially what we've got is one molecule going to a protein or an enzyme and being broken into two different molecules. Now, here what we're seeing is an input, and that one molecule, if you look at it, it's the difference between this starter molecule and then these two products, it's really subtle. And what this is showing is that this R sub 2 right here has changed a little bit. Here, it's just a single line representing that R sub 2. And now here it's a little bit of a triangle, and here it's the dashed line. What that means is that this molecule now has this R sub 2 either kind of coming out of the paper or coming out of it's like close to you, and then with the dashed line, it's farther from you. So that's something that happens in chemical reactions. Um, and this is an example of enzyme catalysis. Now, we're going to get into cell signaling and ligand binding and structural proteins a little bit later, but for this exam, I just wanted to get to enzyme catalysis and introduce that. Now, first question, aside from my description that I just gave you, what does an enzyme do? Well, living systems use enzymes to accelerate and control the rates of vitally important biochemical reactions. Next up, enzymes are the agents of metabolic function. What that means is that whenever, here I've got a vanilla Coke. When I drink that vanilla Coke, well, there's some sugars in there. My body is going to take those sugars and break them down and take that sugar and convert it into energy that I can actually use. Now, in terms of a enzyme, here is an example of an enzyme. This enzyme is known as lysozyme. Now, lysozyme, the enzyme, is what we're showing here is that it's binding its substrate or ligand. The difference between these two terms is that a ligand is a molecule that is not acted on catalytically. What that means is that, let's say that my hand is an enzyme and this pen is my ligand, or sorry, my hand is a protein. A non-enzymatic reaction would be this pen is my ligand, my hand grabs the pen, and then at some point releases it. Okay, nothing about this pen changed. Now let's switch it up. My left hand. My left hand is an enzyme. This is my substrate. Listen closely. Okay, I press that button. That's a catalytic reaction. Now my hand will release that substrate. So the difference between an enzyme, or sorry, a ligand and a substrate is that a ligand is acted upon and something changes, whereas a substrate is acted upon and something about it changes. A ligand is just bound and released. So we're talking about enzymes, so we're talking about substrates, those small molecules that an enzyme can bind. Now that substrate is going to bind in a particular location on that protein. That location is known as the substrate binding site. That substrate binding site is adjacent to or next to a site known as the active site of an enzyme. Now, to visualize our enzyme and what an example reaction would look like is this enzyme binds to one substrate and then potentially a second substrate. So some enzymes are what are known as bi-substrate reactions, meaning they catalyze bi-substrate reactions, meaning they have two substrates. Now, here what we've seen is our free enzyme and then our free substrate A and our free substrate B. Now we have a complex between our enzyme and our substrate A and substrate B. This is known as the ES, or the enzyme substrate complex. That's where the two components being the substrates and the enzyme, have come together to form one. Now, in this case, there's actually kind of three components because there's two different substrates. But for all intents and purposes, we can refer to this as the ES complex. So what we've got here is basically E plus S. Here is our ES complex. Then that enzyme brings these two components close enough together that our enzyme changes its shape a little bit and causes a chemical reaction to take place. Causes that chemical reaction to take place between A and B. Our two substrates react with one another. This complex is known as the ES star complex. This is where a reaction is taking place. Our enzyme substrate has the reaction take place. So this is a subtle difference, but that star there, or that asterisk, is super important. When you see ES asterisk, think, aha, reaction taking place. Now, here, there's no asterisk, so no reactions taking place. Okay, next up, our substrates affect binding, or our substrates affect enzymes on binding. Okay, now what's happened here is that our reactants, or our substrate A and substrate B, have undergone a change. Substrate A becomes product C. Substrate B becomes product D. Now, what we're kind of seeing is that part of A has moved to bind to part of B. So this is our EP complex. So essentially, if you think about the reaction taking place, you have the enzyme and substrate forming a complex. Then the reaction takes place. Then the enzyme and product are still a complex. So this complex, ES, ES star, and EP, there's an entire profile of what's happened there. Now, when we are done with this, our product C, what was originally substrate A, our product C is going to be released. In addition to that, product D is going to be released. Now, the enzyme is going to be freed to bind more substrates at this point in time. This is where the enzyme and the substrate, or and the products, are separate from one another. E plus P is what denotes that separation. So the progression of what's taken place here, our enzyme and our substrates are in the same area. They're not in contact, but they're in the same general vicinity. Then we have our enzyme substrate complex, where our substrate binds to that enzyme. Then, after that binding has occurred, our reaction takes place. Sometimes people like to just characterize this and write chemistry, essentially saying, hey, a reaction's taken place, some chemistry went down, done. The last part of this, before our products are released, is where our product formation has occurred. So substrate binding, chemistry, product formation, release of the products. Now, when we look at these what's that? 5 different displays. What we have,
Well, anyway, it looks like it's full.
what we have here is we've got 1, 2, 3,
Yeah, your sheet, the
4,
one you posted is
5,
less robust.
5 different kind of steps or phases in this catalysis. 1 and 5. What do they have that's different and distinct from all the other ones? Well, that's the free enzyme. Only then is the enzyme able to bind to something. Now, an important distinction or an important thing to look at with all of these is these arrows right here. One thing that these arrows denote, and to put it, I'll rewrite it on a line, E plus S. There we go,
Oh, I think so.
ES. Some people will also write this like this. These two lines, or these two, are distinct from that. Because what this says is that this reaction is reversible. Let me cover that up. Let me erase that. This reaction is reversible. Whereas this is going in a single direction. This is almost like a one-way road. This is a two-way road. This is a one-way. So those are distinct and different from one another. Now, this is what we can do with this information is we can classify different types of reactions. For starters, we'll look at a total of 1, 2, 3 reactions. These 3 reactions are, I would argue, simple and straightforward. Because if we look at reaction number 1, you have one substrate making one product. Number 2, you have one substrate that leads to the formation of 2 products. The way that I always like to think about this is, let's say that molecule A is a box. Or actually here, we'll use some organic chemistry terminology. Let's say A is this box. And somehow this would be reactant A, product B would just be 1, 2, 3, 1, 2, 3, 4. So this is 4 carbons. This is a cyclopropane molecule. And this is a linear propane molecule, or butane. Cyclobutane here, a 4-membered ring. And then here is a linear 4-carbon molecule. Now, a single substrate going to multiple products. Well, here what we could do is our input could be, or our A could be 1, 2, 3, 4, 5, 6. We've got a hexane.
So,
What would give us 2 products of that? Well, we could have a ethane, 2 carbons, and then 1,
no,
2,
I'm right
3,
there with you.
4. A 4-carbon molecule. So we're going from 6 carbons to 2 carbons and 4 carbons. So we're splitting that molecule up effectively. Our multiple substrates making a single product would be 2 carbons, boom, plus our 4-carbon
Yeah, but I
molecule.
don't think we
4
are.
carbon.
Now I think actually.
And that would give us our 1, 2, 3, 4, 5, 6, our 6-carbon molecule. Okay, so that sums up our first 3. Now, if we look at multiple substrates and multiple products, it's a little bit more complicated. But when we look at those different reactions, we can classify them a little bit. We can classify them as an ordered reaction, a random, or a ping pong reaction. And what we're going to look at with respect to these are the models and the mechanisms for these. So here what we have is substrate addition and product release. So What we're doing now is we're just looking at these right here. Multiple substrates going to multiple products. Okay. Now, the order of substrate addition and product released in most enzymatic reactions follow two reaction mechanisms. They are sequential, so the order of substrate addition and product release. Okay. Now, just to give you a little bit more context,
Okay.
this right here, essentially what we're asking is which binds first, A or B? Or does it matter? That's what we're investigating when we're looking at multiple substrates and multiple products.
Well, Paulina just posted
Okay.
this one.
So a sequential reaction is a reaction that says all substrates must bind to the enzyme before the reaction occurs and products are released. Okay. The sequential reaction can either be ordered or random. Now, in an ordered reaction, well, then the sequence matters. A random reaction, it doesn't matter. So if we were to look at a sequential reaction, I'm going to jump back in just a little bit, but a sequential reaction is all substrates must bind to the enzyme before the reaction occurs and products are released. Now, this graph right here, this diagram right here, shows E, your free enzyme, binds substrate A, just like what we looked at earlier. Substrate A and forms an EA complex. B then binds to EA, making an EAB complex. This right here is essentially ES. ES can then go on to have the ES star. That ES star can then go on to form EP, which in this case is EPQ. Okay. So what this shows, and the most important takeaway from what I've shown so far on this slide, is that A must bind before B can bind. So what that means, or another way of looking at it, is in this model, as I show it, EB will never form. And the reason for that is because EB cannot form. What that means almost is that A has to bind to E before B can bind to make EAB. Okay. Now, as soon as EAB forms, then EPQ, or the two products, can be generated. Now, this sequence also matters. The first product, P, is released, and then the next product, Q, is released, giving us back our free enzyme. So we start with our free enzyme and we end with our free enzyme, just the same. Now, this is, like I said, the ordered sequential reaction. This is a sequence that it has to follow. EA can form, EB will never form, because in order for B to bind, A has to be present. That is an essential part of this reaction. Now, next up, a random sequential reaction. This shows no preferred order of substrate addition and product release. In this case, our enzyme can, let's say that you have two copies of the enzyme. One will go in this direction, and the other will go in this direction. It does not matter. So what that means is that in a random sequential reaction, EA can form and EB can form. It's almost flipping a coin. Which direction is it going to go in? Now, when EB forms, well, then you're going to bind A next. And as soon as you bind A, you'll form EAB. And then your reaction, your chemistry will take place, and EPQ will be produced. And that EPQ, well, you're going to release your first product in one situation, in one scenario. Then you're going to be left with EQ, and then Q will be released, and then you'll be left with E. Whether you're going in this direction right here or this direction right here, it's totally flipping a coin. It does not matter. It's one or the other. Both can occur. Both can occur in the exact same mixture. Now, what that means is, or that word random is very important for understanding this. You've got your ordered sequential reaction and your random sequential reaction. The next situation is known as a ping pong reaction. In a ping pong reaction, it gets a little bit complicated. So we'll read this and then we'll process it for a minute. One or more products are released before all substrates have been added. Okay. And an alternate substrate enzyme form F is produced in the half reaction. So essentially what that means is this is like that ordered sequential reaction. So like ordered like the ordered sequential reaction. E binds A,
So, Carrie, do you think this is
producing EA. Okay. Now, this is where, oopsies, this is where our chemistry is going to take place. Notice how on this slide, we bound both of the substrates and then chemistry occurred. Here, we bind one substrate and the chemistry occurs, which gives us our first product. Okay, so I'm going to do some erasing. A becomes P. Got it. E becomes F. Wait a minute. A becomes the new product. That new product is the first product that's released, P. Enzyme becomes F. Okay, that enzyme that becomes F, that F is the, man, that's kind of wonky. That's kind of crazy. That F is the enzyme. And this is the modified form of the enzyme. That modified form of the enzyme is able to bind the second substrate, B, making FB. Okay, so that B is our second substrate. B becomes Q, our second product. F, our modified form of our enzyme, returns to the original form of our enzyme, E, and Q is released. And we regenerate our free enzyme. Okay, so essentially what this shows is that, oh yeah, and let me add in this detail over here. So between our sequential reactions and our ping pong reactions, our sequential reaction has, well, a designated and an essential and a necessary sequence. Our ping pong reaction does as well. Our random sequential reaction, there's no essential sequence that has to take place. Whereas in our ping pong reaction and our ordered sequential reaction, there is. Okay. Next up, one thing that distinguishes a sequential reaction from a ping pong reaction is that in the ping pong reaction, there are two steps of chemistry. Whereas a sequential reaction, there's only one, whether it's a random or an ordered sequential reaction. Now, I always like to think that these are kind of, let's see, what's the best way to, they're kind of simple from like a conceptual standpoint. A ping pong reaction, this modified form of the enzyme, it's almost like the enzyme levels up. It picks up something along the way. So I always like to think in terms of metaphors and examples. So the enzyme here, it's almost like it learns some skill. And where does it get that skill? It gets that skill from our first substrate. That skill is picked up by our free enzyme, and then it releases our first product. Now, that modified form of our enzyme, the enzyme with that new skill, then is able to pick up this new substrate and pass along that skill to that new substrate to generate our first product. Then the enzyme is basically recovered and it goes back to its original state. So one other way that I always try to think about this is I like to think about complexes that can form. Can EA form? Yeah. What about EB? Can the EB complex form in a ping pong reaction? No, it cannot. The EB complex cannot form in the ping pong reaction. Why not? Well, because we have that modified form of our enzyme. Only FB is going to form. Okay. What about an FA form? No, it can't. Since FA cannot form, or the reason FA cannot form, is because FA includes our modified form of our enzyme. So all of this, like the important takeaways from all of this. So in preparation for your exam on Tuesday, related to this material. This right here is made up of amino acids, tons and tons of amino acids. These molecules, or this enzyme being made up of amino acids, well, it's got a primary, secondary, tertiary, and probably a quaternary structure. What kind of interactions stabilize those different structures? Well, there's covalent and non-covalent interactions. What types of interactions attract this enzyme to this substrate? Well, covalent and non-covalent interactions. All of these different types of interactions are what attract these two things to one another. Now, is that to say, like, that's why we care about amino acid structures. We want to know what types of forces attract amino acids to one another. And we also want to know and are curious about the forces that attract amino acids to substrates. Because if we understand that, then we have a good way to design drugs. We have a good way to understand interactions at the molecular level so that we can, learn about how to cure diseases. Okay, well, like I said, you have your exam on Tuesday. That exam is going to open up. Sorry, the exam opens at, let me go back to my beginning slide. The exam opens at 1:30 p.m. You'll be taking this exam with Proctorio. It closes at 11 p.m. And if I remember correctly, nobody really encountered any issues that prevented them from taking the exam. If you have encountered an issue and you haven't resolved it, please contact me so that we can resolve it before Tuesday. I want this to be like a seamless experience for you. So your exam opens at 1:30. You have 80 minutes for it. And like I did last time, I'm going to kind of make a video where I review all the questions and scrub through it, make sure that it's all good and that I don't have anything marked incorrectly. I know that last time I did have a question that I just kind of breezed through. Hopefully that won't happen. But I want you, like, in preparation for your exam, like I can't stress this enough, do the practice exam. After the practice exam, find the questions that you missed. Why'd you miss them? Try to diagnose that. And then after that, I would look at that old video that I made where I walked through exam number 1 and went question by question of that. Make sure that you're good on the exam 1 material, just like you're good on exam 2 material, because exam number 2 has old stuff on it for 5 of the questions, and then about 20 questions that are new stuff. That's all that I had. I'll go ahead and keep recording for a little bit if anyone has any questions. But if nobody has any questions, I hope you guys have a great day. I'm gonna finish up the grade reclamation in just a minute or just a few, hopefully before the end of tomorrow. But other than that, I hope you have a great day, great weekend. Email me if you got any questions. Like I said, I'll probably be on for office hours tomorrow, and I will, yeah, just have a good one.